So further to my previous video on researching plants in native habitats of leopard gecko, and I started to talk about the electrical conductivity of the soil. Now this device here is called a soil quality sensor, manufactured by IBBot. And it can be synced, so the information, the data from this can be synced to phones and laptops through Bluetooth. Now basically what this measures is soil moisture and soil quality. I'm going to just give you a quick demonstration. So when you take this cap off, you can see the sensor probe there. When that gets dipped into the soil and left for a bit of time, the data will then be synced to this screen on here and on the app on my phone. And basically soil moisture, so that measures the amount of water that's in the soil at a given time. Now the last measurement was taken at 4:30 this afternoon, and it was on the warm side, and it measured 70%, which means that the soil there is moist, quite moist. So there is a fair bit of water in that soil. Now, electrical conductivity, or EC, is a measurement of electrical ions within the soil. And the ions are dissolved through the process of the water moving through the soil and dissolving salts through processes such as osmosis, which releases nutrients into the soil. So the EC basically measures the amount of nutritional salts which have dissolved from the water and placed into the soil. And the EC is measured in millisiemens per centimetre. And so we can see here that the last measurement at 4:30 this afternoon on the arid zone, the EC reading is 0.9 millisiemens per centimetre, which means that the salt levels in that area is relatively rich, which suggests that the nutrient content of the soil, for example, sulfates, which is salts, nitrogen, carbon, is present in the soil in the arid zone. And this links to pH values, as certain plants, well, all plants have their own needs and pH preferences. So we have acidic soils, which pH colours red, orange, yellow. Then neutral pH is usually a colour green, and then alkaline pH is signified by the colours blue, violet and purple. And further to that, I did a little test on pH. So these little test tubes I bought from my local garden centre, they were £1 each, and basically they were supplied with a capsule of powder, which is the residue at the bottom of the tubes, So you insert the powder into the tube, and then above the powder you insert a sample of soil from about a few inch depth and place it into the tube up to the first line, so roughly so about there, and then you top that up with tap water to the last line, and then you give it a little shake and then let it settle, and it will reveal the pH colour of the soil sample. So the left tube is a sample taken from the arid zone, where the soil is more clay and topsoil, earth mix arid, and the tube on the right is taken from a sample from the woodland zone, which is more a mixture of orchid bark mulch, topsoil, organic topsoil, vermiculite, sand and sphagnum moss. And then we can see that the soil in the arid zone is slightly more alkaline because it's more sort of green in colour, neutral to alkaline. And then on the right, so in the woodland side, it is more acidic because it is swaying from green to yellow. And the plants which are in the terrarium all have preferences for neutral to slightly acidic or slightly alkaline pH values in the soil. So then we can infer from the test results that the quality of the soil is suited to the plants that are in there. So in theory, this should help optimize their growth. And referring back to the EC measurements, the EC measurement taken from the warm side suggests that there is a quantity of salts and nutrients within that soil to optimize plant growth, and we can see that visibly by this aloe which has taken off. This aloe started off as a small little plant about that size and has now grown, and have even propagated from that plant and transplanted into other areas of the terrarium. So that shows that the nutrient content with that soil is rich. And also the biodiversity within that soil, so like the springtails and isopods, are very active. Similar to the woodland side, where yesterday I figured that there was a load of springtails and isopods in the soil after digging some of it out, because I was wondering why my plants initially on that side were dying, but it was because they were planted in shallow soil placed above chunky logs which were buried beneath the soil to form burrows. However, due to the logs, they were obstructing root development for the plants. So therefore I moved the logs and rearranged them to form like a tree trunk area in the back, and then placed a new substrate using vermiculite and sand and organic topsoil and sphagnum moss as the top layer of substrate, and then planted directly some new plants directly into this substrate.